Good morning. My name is Eric Aliwa, and I'm the President and CEO of PDCline Pharma. I'm going to present you over the next 10 minutes the new class of cancer vaccine based on an off-the-shelf presenting cell line we have developed within the company. So in a nutshell, the company is a spin-off from the French Blood Bank. The scientific founder of the company, when he was an employee in academic research lab associated to the French Blood Bank, has identified in the blood of a leukemia patient, a very rare leukemia affecting the plasmacytoid dendritic cells, which is the small fractions of the circulating dendritic cells in the blood. And this leukemia was affecting this cell. And based on the blood of his patients, the PBMC, he developed a GMP cell line and a master cell bank approved for clinical use by the French authorities that we are still using today as a vaccinal platform. So today we are a team of more than 25 people, including seasoned executives from biotech and pharma. In terms of funding, we get close to 32 million euros in equity and non-dilutive money. Our proprietary PDC line has three key advantages. First of it, it's a cell line, which is extremely easy to expand in bioreactor. I will show you that later. The origin of the cells, being a plasmacytoid dendritic cells, provide very interesting potent antigen presenting cells capabilities. And last but not least, it's a very versatile platform. We can load the cell line with tumor antigens representative of any kind of tumor, solid or liquid. So it's broadly applicable to any cancer. We are clinical stage. The first phase 1b academic feasibility study in melanoma has been completed and published. And we have now ongoing a phase 1/2 in non-small cell lung cancer. The pipeline is summarized here. We have the PDC-MEL, the melanoma with 4 shared antigens is completed. It's not active anymore. The result has been published, as I said. We are focusing today on the PDC lung with the 6 shared antigens and on another modality, which are the neoantigens based on the tumor mutations that is in preclinical stage. One important element to mention is that we have signed a first licensing deal with a South Korean conglomerate, LG Chem, which is a huge organization. LG Chem is the 10th chemical industry in the world, and they have a growing life science department, and we signed a licensing deal with them to develop PDC lung cancer vaccine in Asia. So they have exclusive license in South Korea and options in other Asian countries. The total deal value is $123 million plus tied royalties on net sales. The company has as well an important asset, which is the management team. We have very competent and experienced management team. We have, for example, Shanna De Bruyne, which is coming from GSK Vaccine, our medical director. She led the registration trials of the GSK MAGE-A3 cancer immunotherapeutic in lung and melanoma. Claude Dedry, the VP of Pharmaceutical Operation Quality, has a long experience in cell therapy and QC control, and we have the 2 co-founders with me of the company, Joël Pluma, the scientific co-founder that joined full-time the company, with an immunologist and the inventor of the technology, Laurent Lévy, which is the CEO, CFO that joined as well full-time the company, and finally myself, which is a serial entrepreneur. I've co-founded several biotech in the past, and I've done several exits in terms of IPO and M&A. We have important to mention a very interesting and competent board of directors. We have as a president Monder Majoubi, who is the current CEO of Innate Pharma, but he was as well the senior VP of AstraZeneca Oncology, of Genentech, and other big pharma. We have as well Jean-Paul Prils, that was the former global R&D director of GSK Vaccine, who is an independent board member and investor in the company. And we have Alain Herrera, an oncologist and the former business unit head of oncology of Sanofi. We have as well a representative of the investors, the Belgium and South Korean investor, because we have in the last financing round the arrival of Korea investment partner, which is a very large private equity and VC fund. The company achieved very significant milestone over the last 24 months. We have in January 2019, we get the accreditations by the Belgium Federal Agency for the QC and release activity of PDCline. We signed in March 2019 the deal with LGChem I mentioned earlier. In June 2019, we get the authorization to launch the phase 1/2 in lung cancer in Belgium and France. We get the approval in Germany in December of 2020. Shanna De Bruyne, our medical director, arrived in August. And in December, we closed the 20 million B round of financing led by the multi-billion Asian VC Korea investment partner. We administered the first patient with the cancer vaccine in February, strengthened the board of directors with the arrival of Monder Majoubi, I mentioned earlier, in June, and dosed the first patient with the high dose in the cancer vaccine in December. So the company has been quite busy over the last 2 years. In terms of technology, how it works, as I said, we have a master cell bank, GMP approved, where the cell line is in bag in liquid nitrogens. And we have developed from this master cell bank working cell banks. And from the back of the working cell bank, we can seed a single-use bioreactor, and the cells will grow in suspension in a synthetic medium where it's serum-free. And after 9 to 10 days of culture, we have billion and billion of cells that are produced. And at the harvest, we separate the cells according to the number of peptides we have to load, and we load individually the cells with the peptide, as you see here, the yellow, red, green, and blue. Then we pull the cells and irradiate to stop the proliferations. Then the cells are put in an off-the-shelf product ready to use. It's a technology from aseptic technology and kept in liquid nitrogens. And we just have to ship to the hospitals. It's put in minus 70 or 80 degrees fridge. And once the patient is ready, there's just to tow the system, the vial, and after with a syringe, take the cells and infuse to the patients. So the manufacturing is extremely scalable. The batch-to-batch consistency is extremely high with this current system, and the cost of goods is, of course, quite low. So we did as well different benchmark study to demonstrate the potency of our cell line. As you see here, some results in vitro, where we have loaded the cell line with a specific antigen, which is melanoma, and put in contact with PBMC, a white blood of donor. As you can see, we have followed the fold expansions of the CD8 T cells specific, the cytotoxic T cells specific of these antigens in the blood of the donor. And we compare with conventional approach, which are the autologous conventional DC or the allogenic conventional DC, which are the myeloid one and not the plasmacytoid. And as you can see here, by FACS dextramer, we see that with PDCline, we're 20 to 200 fold more potent. We did study as well with neoantigens, where we have selected 2 neoepitope where the mutations in the peptide identified in the blood of cancer patients was clearly identified, what you see here in red. And we put both peptide mutated in contact with the blood of healthy donors. And it's a CD8 T cells purify. And as you can see here from the 3 different donors, HD01, HD02, and HD03, you see by FACS up left and bottom right that in red and purple, we are definitely able to expand the CD8 T cell specific of each peptide. We have reached a range of 150-fold increase. That despite its healthy donor, where the concentrations of specific CD8 T cells is extremely low. It's 1 out of 1 million in this specific setting. So, as I said, we have as well completed a academic phase 1B clinical trial in melanoma on 9 patients, where the cell line was loaded with 4 melanoma antigens, which are tyrosinase, GP100, melanase, and MAGE3, sorry. And the result was definitely promising. We get acceptable tolerability and safety profile. We didn't observe any sign of rejections. due to allogenic rejections, no anti-HLA antibody detected. And we have observed significant antigen-specific CD8 T cells directly detectable in the blood of patients in some of the patients of the study. And these results are very interesting because we get them despite we infuse just 3 times, which is not the optimal way. We will infuse 6 times in the next clinical trials. And we didn't co-administrate any checkpoint inhibitor. But we know that there is a synergetic effect between our vaccine and checkpoint inhibitor. To demonstrate this synergy, what we did is we have selected the blood of melanoma patients. In melanoma, there's very high tumor-induced immunosuppressions, and we put the PBMC of these melanoma patients in contact in culture with the PDC line loaded with melanoma once again. And after 1 week of culture, we follow by FACS dextramer the fold increase. And as you can see here with the PDC line loaded with melanoma, we have 15-fold increase compared to baseline. With the checkpoint inhibitor alone, we have two times fold increase, which is not statistically significant. But if you combine both, it's where you observe this very nice synergetic effect. We have 35-fold increase due to the mechanism of action that are completely complementary between both modalities. So we are now conducting, as I said, non-small cell lung cancer clinical trials. We have selected 6 different antigens, comprehending MAGE, multi-MAGE, MAGE4, MAGE3, NY-ESO1, MUC1, survivin, and we have loaded this cell line with these different antigens. We are, in fact, enrolling 64 patients in this study. The main objective is the safety and demonstrate immunological activity and clinical response of the PDC lung, the vaccine associated with checkpoint inhibitor. And we have 4 cohorts of patients in this study. In the 2 first cohorts, it's in adjuvant setting after chemotherapy, where we inject the vaccine alone, low dose first with 6 patients, then high dose with 10 patients. Then we pass to the low dose in first line metastatic non-small cell lung cancer patient in association with checkpoint inhibitors, 6 patients. And then we go to the high dose in the same setting, but with 40 patients. It's this last cohort we would like to observe a clinical response. So we'll infuse 6 times, this time weekly infusions, and it will be IV and subcutaneously infusions both. Just to finalize my presentations, the timeline and the funding strategy, we completed a 20 million euro of financing to finance the current clinical trials. We expect to get interim clinical data Q3 of 2022 and to get the final report of the current clinical trials in Q1 2023. At this stage, we could envisage a new financing, an IPO or a global deal with the pharma companies. And I would like to thank you for your attention and to finalize with a nice picture of my team. Thank you very much.